I came to research through a love of knowing how things worked. So I did a science degree here at Sydney University and got to play with microscopes and laboratories, chemical labs and biochemical labs. And I just loved the idea of working with my hands, but also seeing how life worked. So I chose to do a research project, what at the time we call honours. It's a 1 year research project. I got to play with the bigger toys, things called chromatography systems and incubators and all sorts of equipment, and really loved it. But the best part was figuring out how cells work. So I spent that year on the most obscure bacteria known to the human race. And I discovered why a mutation was causing it to, I guess, function differently, become pathogenic. But it's the process of learning how life works, of this. It's the Star Trek thing, going where no one has gone before, learning things that no one has ever seen before. That's how it started. So then I chose to do a PhD in something other than bacteria. So I switched to a PhD program, studying proteins in the brain. I'll talk about proteins later. Proteins that control nerve communication, how two nerves talk to each other. Basically, how does our brain work at the cellular level? And I thought that was just the most exciting thing in the world, to be part of that. Just the nuts and bolts of how do the nerve cells that make up our brain, how do they talk to each other? How do they communicate? So we can describe it. We can describe electrical activity of nerves. We can describe how nerve conduction works, basically voltage. We can describe the physiology, but I wanted to understand, but how do the cells themselves work? How do they make an electrical signal? How do they receive an electrical signal? And that's happening inside the cell. It's so cool. And the point was, I got to play with all the toys. I got to be hands-on in the laboratory, growing cells in culture in a dish, working out how to answer the questions I wanted to answer. At the same time, it's in a bigger picture context. Over time, the work could contribute to human health. And that's sort of the underlying 10, 20, 30-year goal. Right now, that's where I'm at, working on that space. But research is about, at the time and still, trying to explain biology, in my case, trying to explain the inner workings of cells. And I started with nerve cells. And it's like detective work. It's like piecing together data that you know, the facts that you know, making projections, predictions about, well, if we know this, maybe this calcium influx, maybe this protein interaction is part of explaining how nerves respond. And then going away and testing those ideas. So research is about a voyage into the unknown. So at the end of that PhD, what I learned was there's a protein, one protein of thousands, inside nerve endings. Every nerve ending, it turns out, in the world, including flies and animals, that one protein responds to a nerve stimulus faster than any other. And I gave it a name, and I've spent my career on that protein. At the time, we named it D-phosphin, and now it's known widely as dynamin. But to be able to be part of finding that it's the fastest response in nerves at a protein level that we know of then and today, only completed that project 40 years later, this year, completed that project of how it responds so quickly and why it responds so quickly and what it does. But back then, all those years ago, was the excitement of the discovery that there is a protein, one of thousands, in a nerve cell primarily. And in a nerve cell, it responds to an electrical stimulus incredibly fast, right? What we call sub-second kinetics. So now we know it's 10 milliseconds, so 10 hundredths of a second it responds to a nerve stimulus. And over the years since, it was trying to figure out how and why. So all this added up to me to a love of proteins, a love of understanding the molecular events happening inside our cells, not just nerves. And that's because this protein that I found during my PhD turned out to be what we call phosphorylated or modified on this fast time frame. And that led to ideas of, wow, if we can drug that protein, if we can design a compound that could one day be a drug to modify that protein, maybe we can modify nerve communication itself. Why would we want to modify nerve communication? Because of things like seizures, you might want to turn down nerve communication. Because of things like schizophrenia, you might want to turn up nerve communication. And I had found the protein that one day I believed could be the guy that could turn into a new medicine. 40 years later, yes, that's what we've achieved. So science is a long road. Science does not always have happy endings. It has lots of blind alleys, lots of failure stories.
But how do you define failure? Or is failure a bad thing?
Failure in science is very difficult because it depends on your perspective. So for a student or for a younger person or for someone looking for a grand application, failure means that in the short term, in 1 year, 2 years, 3 years, you haven't succeeded and you're not going to get noted, recognized, you're not necessarily going to get a grant, you may not even get your PhD. But failure from a scientist's point of view is a learning exercise. It's, well, we know that's a blind alley. Or more than that, it's a new fact, and how can we modify our ideas to fit the new fact in? And that leads to whole new ideas. It's, it's back to square one on that particular question. How do you reframe the question? Was I asking the wrong thing in the first place? What do I have to do to redesign my experiments, my approaches? So in the big picture, failure is a really important part of science. It's a necessary part of it. So long as you can pick yourself up, shake yourself off, pull the team together if it's a team, and say, well, what does fit the data, especially now we have these new facts? There's a recent failure that sticks in my mind, and it's a student in my lab at the moment. This is an aside, but students are the driving force in labs and postdocs because they just go over the top. This project was about a protein called dynamin and an idea that a future drug would be binding to the surface of dynamin to block its function at a very specific site. We even gave it a name, Site 2. And the students' project was, with me, together, to test the hypothesis that the drug bound to Site 2. And it's not a drug yet. It was a research chemical that we want to turn into a drug. And for 3 years, it's like nothing worked. So we tried a different approach. Then we tried another approach, completely different methods, different ways to tackle the same question. Does the drug bind in that spot? We mutated the spot, still binds. We measured the protein folding, it still folds. So nothing made sense until we came to the realization, oh, the hypothesis was wrong. It's not binding there. The data adds up overwhelmingly to show the hypothesis is wrong. It's not the binding site. So the student, again, at this point, at first was a bit depressed. The project is a failure, but it's not. Because what it means now is the team is looking at, well, what is the mechanism of this compound binding to dynamin? And the mechanism must be something else. We're looking at the data wrong. We're looking at the evidence, the facts that we have. We're looking at it through thick glasses instead of open. This experiment, this one didn't make sense then. Oh, now it does. That's not the binding site. It's doing something else entirely. And why that's important is because that allows the project to go forward. So there's a failure in terms of a couple of years. And that couple of years, it's a side trip that's led us back to what's actually happening. So that failures are major parts of science, but on a small time window, they can be devastating for the individual involved if there's funding or time limits for research, or study programs have to end. That can be devastating. But for the project itself, no, they very frequently they turn into something positive. If you think about it, you've got to put the pieces together, re-look at what you know from fresh eyes. I use my hand a lot to explain what a protein is. So I'll do a lot of this. So to explain proteins, you have to start with the DNA. You have to start with the genome. So the genome is the list of all of the genes in our body. So in any one human, one person, the genes in our body are the same in every cell. Every cell, every part of our body has the same chromosomes where the genes reside, the same DNA. And what DNA is for, in big part, is codes for proteins. So those codes are called genes. So there's around 20,000 genes in a human genome. Interestingly, a frog has 44,000 genes. Why do they have more? Humans have 20,000 genes, and they're pieces, like this piece, that piece, that piece, of the long DNA strand that make up our chromosomes or make up our genome. So for proteins are what genes do. They make proteins. And proteins are why one cell or one tissue is not the same as another. So the DNA in my skin, the DNA in my heart is the same. It's exactly the same DNA, same genes. But those genes are expressed, they're turned into proteins. And the ones that are turned into proteins that make skin are different to the ones that turn into proteins to make heart tissue. And that's true for all tissues that exist. There's hundreds of them and all species. So the proteins are made from the code of the DNA. So each gene represents one or many proteins. So what are they? So it's hard to explain a protein as a string, but it's a string of amino acids. And these are chemicals, very simple chemicals. There's 20 kinds. They're strung together in a straight line. And the gene is what codes for which amino acid goes in which order to make a protein. But proteins are not strings. DNA is a string. Proteins are three-dimensional folded machines. So that string of amino acids folds into shapes, almost unlimited numbers of shapes. So some of those shapes bind together in long strings of proteins now, and they're structural. Proteins like tubulin and actin, they form the walls and the structure and the architecture of a cell. It's shape. And other proteins fold into three-dimensional shapes, well, all proteins do, with pockets. And those pockets might be what we call enzymes that convert one chemical in our body to another to make energy, to make, for example, the energy molecule of our body is called ATP. So there's an entire set of genes in all cells in all of life that exist only to undertake a series of chemical reactions to convert chemicals in our body bit by bit into ATP, the energy molecule. And there are other proteins, like our muscle proteins, that use that ATP, binding to different parts of those proteins, and break it down, releasing energy for their function. So proteins can be enzymes, that is, converting one chemical to another. Proteins can be structural. Proteins can control the way DNA divides, the way our chromosomes divide when cells grow and divide and turn into two, and basically people grow and tissues develop. So the genome in all our cells is the same. The genome in your cells and versus my cells will be slightly different depending on our heritage and our families. But the proteins that they make determine the tissue type, but they also determine the state of the tissue. So a really good example is red blood cells. There's a protein that is made from our genome that lives in red blood cells that binds molecular oxygen and delivers it to our body, hemoglobin. But the protein that's used in embryos and used early in childhood is one form, and that changes. The body says, I'm going to switch off that gene and I'm going to turn on a different hemoglobin gene for an adult with different properties for binding oxygen. So proteins change, even though a red blood cell is still a red blood cell, right? It's still got hemoglobin in it, but the one hemoglobin that's there is now different. So when you talk about then the state of a cell, aging, maturation, or a cell turning into a stem cell or turning into a kidney, liver, muscle, the proteins are what makes that change. The DNA controls the change in protein expression or the protein list in a cell, but the proteins that are in a cell at any one time determine its age, its response to infection, what tissue it's going to be. And we call that list the proteome. So the genome is the list of all genes in our body, but the proteome is the list of all the proteins in a single cell or tissue at any one point in time. The proteome is the list of all the proteins in any tissue at any point in time, and it changes with time, and it changes with tissue, it changes with disease. So there's lots of implications of what that means, but one is food. To digest food, to eat food, one of the main things in our food is protein. So when you eat a steak, when you drink a protein shake, you're taking in proteins and you're breaking them down in your digestive system and you're absorbing them. And what we get from that is the individual amino acids, so our cells can build our own protein the way our body wants it to look. But there's many other reasons for eating food, not just that. It provides energies from the sugars, from the fats, and from the proteins that breaks down, that allows our cells to make, through proteins, to make energy and store that energy. So, for example, a steak is not just protein that we're getting, it's primarily protein because it's primarily muscle, and muscle is primarily protein. But there's a lot of other metabolites and bits and pieces in there that the body uses or rejects and throws to waste. The body selects through our digestive system what parts of the food we're going to keep and use, the amino acids and the metabolites, and rejects part of the tissue which we excrete to waste. The proteins are the basic building block of functioning living cells and tissues. But you can't separate proteins from the DNA, which codes to make the protein, the blueprint. So if you control the DNA as well, you can change what proteins are being expressed in any tissue at one time. So the proteins are the most important player, only from a biased point of view of a person who spent his life studying proteins. But they're not the be-all and end-all, but they're the predominant things that you see when you see a physical organ or animal or plant. You're seeing the proteins. The DNA is the blueprint for those proteins. One is a mutation, something going wrong, radiation or a cell division goes wrong and changes the DNA for one gene. The protein that is made from that gene is the thing that conveys the damage, because it can be faulty or it might even not be produced. That's what causes disease. A mutation in a gene leads to a defective protein or no protein at all. That's when tissues go wrong. Many diseases are related to defects in proteins. Cystic fibrosis is one, and that's a channel in your lung that creates fluid, and that channel is made, the cells still have it in your lung, but it's in the wrong location. It has to be on the surface of the lung when it's not there. So the DNA mutations cause defective proteins or no proteins. But the consequence of it is very, very different to what you might think. You can have one protein that's wrong, there's too much of it in a cell, we call overexpressed, or too little, underexpressed. That could be from environment, from a disease, from an infection, virus, radiation, environmental damage. There can be more or less of that protein, but the consequences flow on. So if you have one mutated gene, it doesn't cause mutations in all the other genes. It tends to be one. There are exceptions to that. But if you change one protein in a cell, have more or less, or have a broken version of that protein, the consequences to that tissue are huge. The whole proteome can change. Tens to hundreds and even thousands of proteins will change their balance, the amount that is in that one tissue, because there's one defective player. It's like an alarm going off and a 3-alarm fire, and everything's in chaos. And that change is affecting the whole proteome, all the proteins in the cell. Many of them are affected just because of one damaged player. And that's really important. Knowing what's changed in a particular disease or in a particular infection, knowing what's changed in the proteome gives us clues to how to treat it. Because it may not be the defective protein that we have to have a drug for. It could be another protein in a network that's changing, if you like, in sympathy with the damaged protein. But that tissue is changed, and we need to find a way to treat that disease, whether it's epilepsy, whether it's anything, cancer, anything. We need to know what clinicians, all a clinician wants to know is, what do I treat my patient with? What can you teach? What can the research teach us? Well, in our case of proteomics, knowing the protein lists in a cell at that particular time in that particular infection can sometimes predict, look, this protein is also up or down, and there's a drug for that. Use that drug. And that works. It's not the only thing that works to help treatment decisions, but it's a really major thing. One of the two, I guess, key career things that I've been doing in research over my whole career has been trying to work out ways to modify, control proteins as future drugs. So drugs are chemicals, very simple chemicals, the most part. And drugs, for the most part, their target in a tissue is a protein. There are some exceptions. But the vast majority of approved drugs, a drug that health experts have shown is safe and effective in humans, are small molecules that can survive the digestive system and get absorbed into the body like food, spread to all of our tissues, and find the protein that they're targeted at, that they're going to block. Discovering drugs is an entire research area across the planet for 100 years. It's an amazing journey, and it's part that I'm proud to be part of. So if you go back to some clear examples, one is penicillin, which was, of course, first discovered basically as a fungus growing in a laboratory because someone didn't use the correct aseptic conditions. And so germs out of the air, fungus in this case, grew on an agar plate in a laboratory. But what the scientists observed was the bacteria on that plate that they were actually studying died. There was a fungus and there were dead cells everywhere. And they realized that there was something in that fungus that was killing bacteria. And we know that today as penicillin. So that was isolated, and the species of fungus is named after that. But it turned out to be one chemical. So, I mean, this is well over 100 years ago. But now, what happens is chemicals like that became a drug very quickly because it was a whole new concept. A drug means experimental chemical that's turned into a medicine, has been modified and adapted by science over decades to improve it and to broaden what we call its specificity. So penicillin doesn't kill every bacteria. It kills a lot. So how do we kill other bacteria? So first, how does penicillin work? It attacks the cell wall of the bacteria. Where on the cell wall? So there's an entire science, and there are thousands of researchers looking at questions like that. So what is the cell wall different in other bacteria? And can we find other compounds from biology, from rainforests, from other fungus, from ocean toxins in sponges and marine snails? Can we find other compounds just by searching that kill other bacteria? And yes, over the years, many antibiotics have grown out of that. What motivates me in research is learning how our cells work, but learning it for a purpose. And my purpose has been to understand the proteins in a cell that separate a nerve cell from a leukemia cell, for example, and to find chemical compounds that are one day going to be drugs to control that cell. So with nerves, I want to be able to turn down the volume, turn down nerve communications to block a seizure. And for leukemia, I want to be able to stop the leukemic cell from dividing so that chemotherapy can wipe it out. And sometimes it doesn't. Mostly it does. So what drives me is trying to work out a way, what is a way to block those functions of those cells that are not good. So that means identifying the components of the tissues, identifying what proteins are in there, hence the proteome is really important. But I also came to this space with a particular target in mind, and that was a protein that I discovered during my PhD called dynamin. And I understood at that time that if I could control that protein, I would have a new path to medicine, both for neuroscience and for cancer and for several other indications. But nothing existed that controlled that protein. So if I could find chemicals that blocked it, then in the lab I'd have this incredible tool to be able to advance my biology and understanding how it works. But over time, I've worked with medicinal chemists, and this is pivotal. The chemists can modify the chemical, the molecule that I want to be a drug, and improve it. So eventually I formed a partnership 20 years ago with a team at the University of Newcastle, led by Adam McCluskey. And his team modifies the chemicals that we first found and changes them little bit by little bit to make them better and better and better. And one day we hope that to be a drug that's approved for use in humans. And that's the stage we're up to now. So that really drives me, the study of understanding proteins, which protein is where and at what time in life, and understanding the structure of that protein, what it looks like in three-dimensional space, and understanding the chemicals that bind to it and how we can harness those to design a drug. And I think as a team with the University of Newcastle and University of Sydney and here at CMRI, we've been able to pull together enough evidence to say we've created a new pharmacology around this protein dynamin and other functions around that system, that we're at the tip of the iceberg now about what diseases we can use that for. And we've done experimental work in animal models to show they are effective in a number of diseases. But we still have to find or design a compound, a chemical, that we can call a drug. And that means it's got to be safe and effective and pass a whole lot of safety tests before it can ever go into a human being for a test. What drives me is understanding how we can harness chemistry, small molecules, to bind to the proteins in our cells, in our case, a specific protein, in order to refine them over time, the way we've done with antibiotics, so that those compounds can turn into drugs. And drugs are drugs only when they're proven to be safe and effective in humans. We only know that they're safe and effective in animals at this point. ProCan is a very large team, more than 45 scientists and students and oncologists working here. And we're working on one concept. If we can identify the proteins that are different, we call that the proteome, between different types of cancer, we may be able to develop a test that can identify a tumor type. And we've done that. More importantly, the oncologists that we work with tell us they need to know we've got this patient with a particular cancer in front of us. Genomics has taken us so far, our experience has taken us so far, and nothing's working. The patient's still not responding. Can the proteome, can the list of proteins give us more information that will help us decide how to treat that patient? And that's what ProCan's goal primarily is about. The principle of ProCan is if we can take the tumour's samples from lots of patients, we're talking groups of 100 to 500, and if we know the clinical outcome of those patients retrospectively, and we can list all the proteins in the tumours of all these patients that have the same kind of tumour and compare them, what we find, what we know, is that there are differences in the total proteome. There can be 100 to 500 proteins that are increased in the tumours or decreased in the tumours of those patients that responded to drugs or resist drugs. So if we take two groups of patients, these patients got the standard therapy and survived. These patients didn't. They got the standard therapy and they didn't survive. And if we can define the protein differences between them, then in the future we may be able to turn that into a test for...
Are you building up a protein library?
ProCan is building the world's largest protein library. We have run more than 28,000 cancer specimens, and we have the list of the proteins that are in each of those tumours. 28,000 samples, but we're seeing around typically 8,000 proteins in each of those tissues with the mass spectrometry or mass spec technology we have today.
And you identify repeat offenders, as it were.
What we identify is the things that the genomics or the mutations haven't told us. We identify proteins that shouldn't be there. We identify proteins that are increased or decreased. And we're learning which proteins from the proteome, from the list, which proteins tell us clinically important information. What kind of tumour it is often an important question, but the most important question is, what can we do about it? Is there, in the list of proteins that are increased or decreased, is there already a known drug? Can we bring out a drug of the 2,600 known clinically approved drugs? Is there already one that targets a protein that's not supposed to be in that tumour? And at a cell level, we know that works. We're trying to do that at a human pathology level and to give that information to an oncologist. Most medicines are small molecule chemicals, so drugs. There are many, many exceptions. There's protein medicines, there's RNA medicines and DNA medicines. They're a small subset, but the vast majority are small molecule chemicals that are safe in humans under controlled conditions and target and kill or block a particular protein or a particular function. So if we can identify what proteins are in a tissue at one time, and there's usually a mutation involved that caused a cancer, for example, we can guide an oncologist to a particular pathway. We can also say, look, here is a set of proteins that are always up or always down in this patient, and there are no drugs for that. And I can go away and try and design one. Another lifetime's work, but that's exactly where we identify a new way that might treat that patient. All cancers can be considered bespoke because the age at which they occur, the organ in which they're occurring, and the environment, what food, what viral status the person has at that time. And we expected that. So if we can take 500 samples of the same tumor type from 500 individuals, the protein list in those people are not the same. The protein list we call the proteome is not the same. Whereas, and their genomes are not all the same. They have variations on a small number of mutations, but they have much larger variations in the proteins that are in those tumors. And we've discovered that by comparing people that survive treatment or don't survive, or people that are going to respond very quickly versus very slowly, we've found that the protein expression, the protein list in that subset can be very, very different. And we expected that. And for every tumor, the differences are not the same. To take lung cancer as one example, we have run several hundred patients with lung cancer. And the very first thing you learn is lung cancer is not lung cancer. Sometimes there's a virus involved, and sometimes there's a mutation involved, and sometimes it's smoking that's led to those mutations. But the proteins in those samples are not the same. The proteome changes in every single individual. And that proteome, that list of proteins in the lung, is different to a healthy lung, always. There's always more proteins in the tumor from one patient with lung cancer and another. And the list of proteins that are changed in lung cancer is as different between individuals as night and day. Within that, what we have to do is sort out those that can be predicted to respond to which treatments. There are many treatments for lung cancer, and there are many where there are complete failures. So the proteins are more different in those groups again. So every tumor is absolutely unique from the protein level. Even if it's the same mutation in the DNA that caused the tumor or is associated with the tumor in the first place, the proteins will still differ from one individual to the next. ProCan is about capitalizing on that data, that information, to make predictions that are clinically useful to guide treatment decisions. Children get basically the same cancers as adults, but they get far less variety than adults. So a sarcoma in a child and a sarcoma in an adult is still a sarcoma, for example. And leukemia in children and leukemia in adults is also a leukemia, and there are many kinds. But what changes is the proteome. So the protein list in a child and the protein list in an adult are slightly different.
Back to the proteins.
Back to the proteins, even if the DNA is the same. So if you could have a mutation in a child that causes a leukemia earlier, or another cancer, for example, earlier, but the same mutation in an adult may not continue to cause a cancer because the proteome has changed in the adult. And you can flip that on its head. As humans age, our proteins change, and our ability to get a tumor from many sources changes. So we become actually more susceptible to tumors as we age, and the variety age occurs. Sometimes protein changes build up over decades, and so the impact of a mutation on the proteome doesn't show for a long period of time. There are many, many explanations, but a tumor is going to grow and evolve in a person according to so many factors, and those factors include age, development stage, and the tissue type where it's occurring. So a different kind of tumor can start at any point in life, in any part of the body. To me, the miracle is this, the miracle that human beings exist at all, life exists at all. Every time a single cell divides properly in an adult or a child, I think it's amazing that it works so many times. We're talking about diseases where it goes wrong. And the fact that it's very, very rare, as rare as it is, the fact that it's as rare as it is extraordinary. Life is incredible.
Well, a metaphor, that's okay. Okay.
Proteins change so much, so often. What I find just simply incredible is that life exists at all. When you learn and discover how cells work, how they grow, how they divide, and why young cells and old cells are different, why children and adults are different, what goes wrong when cells don't divide properly. When you learn all of that, you just marvel at how any cell or any tissue or any organ can function normally without something going wrong. What is also amazing is part of what life is are mechanisms in our cells to protect against things going wrong. And it's when they fail that we get disease. But it amazes me constantly that cells work at all. And then they work together to make a tissue. And then the tissues work together to make a living person. It's incredible. It's just incredible. Shouldn't be like that.
Who's going to tell the big guy?
If I was the scientist designing the system of cells and proteins and genes, it would go wrong a lot more often. We know so little about it. We know so much, but we don't know anything like enough to put one together ourselves. So one example of why ProCan in the future is going to be so important in very many types is a personal story. And a friend who was in her 50s, a very healthy woman, completely hospital-free for her whole life. No problems. One day, back pain. And the back pain wouldn't go away, then scans and x-rays. And within weeks, there was a tumour found, and it was spread right throughout her stomach cavity. And pieces had grown in her spinal cord, causing the pain. Immediate chemo, immediate hospitalisation. Chemo can work very, very well in some people. It just made her worse. And the doctors didn't have any clue about where to go next, what treatments to use next, because the tumour was everywhere, and the identification of where it started was just not there. There wasn't like a lump in her pancreas or a lump on her lung that says, it came from there and spread. It had just spread everywhere. So without that knowledge of where the tumour came from, the doctors were helpless. And she passed away within 3 months from diagnosis, failed treatment, to she passed away. But what if we had the sample, right? What if we were able to see the protein list from the tumour and be able to say it came from the pancreas, that's where it started, or it came from the lung. With that knowledge, oncologists can make decisions about, right, these are the kinds of therapies, or an immunotherapy or a new therapy needs to be used because that's where it came from. We don't know that that would have worked, but it might have, and it gives her oncologist a choice and an option. And we don't know if she would have survived or not. And it's stories like that that drive ProCan. We want to be able to provide the tools, the technologies to be able to empower the oncologist to make decisions on the spot much more quickly. We don't want to send it away to a lab and wait 3 years to get a research question. We want to send it away to a lab and have a result overnight to guide our treatment decision when the chemo was not working.
Okay, you're frying my brain because there's so much information coming into it. It's fun. Steve, do you have any questions?
I got 40 years of this behind me, so
I'm having difficulty keeping up. It's you're telling it really well.
one of the things, is how ProCan came about, but although it's ACRF heavy, it's really about research and funding heavy, if you want to hear that story.
Yeah.
I spent my career studying proteins and discovering compounds that we hope to turn into drugs to control those proteins. And technology has driven a lot of what I do and the research that me and my team and my colleagues do. And one of the biggest parts of that is a technology called mass spectrometry, or mass specs. Mass spectrometry is an old science. It goes way back to World War II, when the first mass spectrometers in the world simply separated two forms of uranium for enriching it. There's a mass spectrometer on the 2 Voyager spaceships, one of which has exited the solar system from way back in the 70s. So mass specs are very old technology. There's 5 Nobel Prizes in the science underneath mass spectrometry. Well, when I first came to this institution, CMRI, 28 years ago, I had to study proteins one at a time by pulling them apart. But in those days, the first mass spectrometers that could measure proteins had been discovered. And suddenly I could identify a protein in a couple of hours, 1 or 10 or 20 at a time. And we were one of the first to adopt that technology, and thousands of others over time, to study proteins, one at a time and then groups at a time. So since those days, and we're now talking around the year 2000 to today, 2024, mass spectrometry for studying proteins, for proteomics, has evolved at an incredible rate. So 10 years later, mass spectrometry evolved with the discovery or an invention that took 10 years to develop of a kind of mass spectrometer called an Orbitrap. And that just changed the face of mass spectrometry more than 10 years ago. And hundreds of these are being sold to researchers around the world because we went from studying 1, 10, 20 proteins per day to thousands of proteins per hour. So that was pretty cool, but it didn't tell us the amount of the protein. Nowadays, skipping another 10 years to where we are today, there's a new mass spectrometer detector inside the same box. And it's after the Orbitrap analyzer, and it's called an Astral analyzer. But the new technology today allows scientists to measure around 8,000 proteins every 20 minutes in one sample. Whether it's a bacterium, a plant, or a human, we can measure huge numbers of proteins very quickly. But the important distinction today is we can get the relative amounts. If we compare 2 samples, we can say these proteins are higher in this sample than that sample. And that concept is what led to the idea around ProCan. What if we could take the tumors from thousands of people, list all the proteins, and know the relative changes in those proteins, we call that expression, in all those tumors, we should be able to use that data to identify what kind of tumor it is and hopefully to understand and make predictions about drug treatment for that specific tumor, personalized medicine for that one tumor. Technologies allowed that idea to grow and develop. So about 8 years ago, we had the technology to do this, but not the money. Mass spectrometers are not cheap. Mass spectrometers are around $2 million each. And to be able to conduct a project on the scale that we envisaged back then 8 years ago required $10 million or more of equipment. And all of our equipment was purchased over the years from competitive research grants. We're completely dependent on our donors and on our competitive grants from government agencies or philanthropy groups. To buy one mass spectrometer is many years of effort. Usually only universities are able to pool their resources to get one or two. So at this time, ACRF came along and offered a one-off, at that time, $10 million award for equipment only. And I was asked by my co-director of ProCan, Roger Riddell. Roger said to me, Phil, what would you do with $10 million of equipment only that needed to be used for cancer research? I said, it's easy. I'd fill a room full of mass spectrometers and I would collect proteomes of human tumours to analyse them for the predictions of cancer type and treatment decisions. Full stop. That was the whole idea. And the thing is, we were funded. So we bought those instruments. We actually ended up by being able to get discounts. We bought 6 of them. And those 6 mass spectrometers at the time had been running for 7 of the 8 years, 24/7, collecting those 28,000 human proteomes from 28,000 specimens from cancer patients for all that time. The world's largest collection of human tumour proteomes on the one platform that's ever been attempted. And we're not even partway there.
Where's the information kept?
So the data from all those proteomes is in one single database, data pool. And we are mining that data for individual tumours to make predictions of patient outcomes from them. But we're also mining the data across all tumour types. Can we make predictions? Can we use the data? One of the things I envisage is what I call a walk-up. So a pathology lab receives one sample from one patient and the pathology request is, what kind of tumour is it? What can you do about it? That's what we envisage. So with the ProCan knowledge base, we're partway towards that. We want to be in a situation where we compare the proteome from one sample that we can collect in one day against the whole 28,000 we have. One day that'll be 100,000, not tomorrow. And be able to make predictions from that we can hand back to the oncologist overnight. That's the goal. The database is many, many other values. There's just huge numbers. We can start looking at cancer mechanisms, which we haven't been able to do before, some parts of it. We can identify proteins that are common in sets of tumours that have no connection to each other. And if there's no drug for that protein, we can encourage scientists like myself to design drugs that might target that protein. So it's an enormous wealth of information, and it's the tip of an iceberg because we're only focused on cancer. What if a program like ProCan extended to all the tissues in the human body and extended from embryos, newborns to adult? And we're part of a bigger consortium that's doing exactly that, based in China, and it's called Pi-Hub. To date, ProCan has focused on what we call retrospective samples, so people who already have a tumour, and sometimes we get the pathologists collect adjacent tissue that is normal or near normal, and we can compare the two. But in the long run, what we really would like is to be able to have a map of how a tumour changes with time. And those changes can be pretty dramatic. But one example is really exciting in this space, and it's come from a study that was led by the UK called the UK Biobank. And what they have is the ability to start predicting diseases, broad diseases, from proteomes collected more than a decade before. So this study is not part of what we're doing here, but it is truly awe-inspiring. The UK Biobank has collected 500,000 patients. Actually, they're not patients, they're healthy volunteers, blood bank donors, collected blood samples every year for 10 years. And that finished 5 years ago. So it's now 15 years since the first sample was collected. The proteome for the first time was analysed just last year on 50,000 of these volunteers. They were all healthy volunteers. But over 15 years, there's 1,000 that have dementia, there's 1,000 that have heart disease, 1,000, I'm making up numbers, 1,000 that have diabetes, and many other human diseases. Yeah. And it turns out, retrospectively, their blood proteome frequently predicted the disease. To take dementia as an example, some patients who acquired dementia had no symptoms of any kind, and yet their proteome from their blood predicted dementia 15 years ahead. And that's happening in a large number of disorders. So knowing the proteome before can be very, very valuable. And blood or plasma from blood is probably the only way you can do that for human medicine, because you can't, what I would like to have is brain samples, but people tend to be using them. So we can't really get brain samples from 10 years ago to compare to a patient with epilepsy or schizophrenia today. So we have to use models. But for blood, it's a really great tool, and it's the tip of the iceberg. It's just beginning in that space. ProCan is not focused on blood samples yet, but we're building that capability. This kind of technology is going to change the world of medicine. We predict from this kind of science a future where a pinprick, a finger prick or a heel prick, collected into a plastic vial, can be stored for decades and can be used then to make predictions about an individual patient going forward. There are a few countries in the world that could do that on a national scale: Australia, Canada, Taiwan, but not very many other places have nationalised medicine. But we just picture a future like that. It's not a reality yet, but there is so much scope there, and that is just so much bigger than what we're able to, I guess, handle at this point in time. But the future of proteomics is incredibly bright. And the funding that we got is what enabled us to be in this space. The 45 or so scientists involved in ProCan would be all having different lives, doing different research activities than this question if we hadn't been seeded by a risky grant or a risky investment in us and the future of this space. So it's really important to convey the concept of how awards of grants that you compete for can actually change your scientific life direction, and of many others, our students. And I think that's really important. It's part of what science is about. Let me put this another way. I got into science as a career because I wanted the freedom to determine my research direction. I wanted to be able to ask the questions I wanted to ask, and I wanted to be able to do the research I wanted to do. I feel incredibly happy, and I've done that, but I didn't control it. The funding agencies controlled it. The agencies that gave me this postdoctoral scholarship versus that one. the NHMRC and other funding bodies that gave me that grant but didn't give me that grant, they guided what I could actually do. I'm still in control, but funding success really also has a big determination of where the future of an individual might be. And it's a really important part about science. You don't know where you're going to be tomorrow. You don't know what you're going to be doing tomorrow. This gets to the question of what motivates me. Science is a motivator because you can go to work every day and never know what the day is going to be. You don't know what you're going to be doing today because you don't know what the data, the results are. You don't know the information, the failures, the successes, and how that's going to change. And on a daily basis, it's new every day. And you don't know what you're going to read in a journal, and you don't know what you're going to learn at a conference. It's a constant quest for new ideas, and that's the passion. Combine that with the ability to impact human health through new drugs or through new diagnostic tests. No, I'm a scientist paid to do my hobby. Just don't tell my bosses or they might cut my salary in half. I heard that somewhere else. Yeah.
Look, there was one other thing that I thought was interesting was the
You know you're off to that one.
yeah,
Hang
it's the battery thing.
Yeah.
Look, actually, why don't we just change sizes? I believe.
Still working on isolating this one protein dynamic. So it's a tremendous
on.
experience.
Professor Phil Tidful. That's the Cincinnati story. Where were we, Steve? You were asking a question?
Yeah, no, I was just asking a question about the tumour and about the fact that if they've been able to determine where it came from. So it's really about
Answer,
why,
Steve, but look at me.
why that happened. Why it's so that in fact, knowing what they know about the tumour, they can't tell which organ it came from.
Tumours are devils. They, what we call sometimes metastasize, and that just means they spread right throughout the body or to different places. So when a tumour spreads from its origin to other parts of the body, it's still the same parent tumour. And unless you know where the tumour originated, you don't know how to treat it. So you have to get a biopsy and do its genome and its proteome to get a clue where it's going, where it came from. So if a tumour is a growth or a lump in a breast or a prostate or something, it's easy then for an oncologist to make informed decisions about, well, we know how to treat a breast cancer, we know how to treat a prostate cancer, and they're not treated the same way. So if they knew that the tumour that was in your stomach cavity or in your spinal cord and everywhere in your body, if your treating oncologist knows where it came from, the tissue of origin, that can dramatically alter the options that oncologist has for treating it. And sometimes it's very difficult to get a sample of a metastasis while the patient's still around. I mean, it's very, very awkward, uncomfortable, surgery unnecessary. So each kind of cancer is treated differently, and there are certain, there's a vast wealth of experiences in how to treat one cancer type versus another cancer type.
But does that mean then that in fact if you kill a tumour in the liver, that the lump that's in the stomach that was as a metastasised tumour would die?
It's frequently the case that if you knew that a tumour that was had spread, metastasized to the wrong part of the body, it's possible that knowing the tissue origin of that tumour would kill the tumour at its site and at its new site where it's metastasized to. There's no guarantee, but there's a higher likelihood that will happen. Nothing's ever black and white in cancer. So a really interesting view of cancer is that today oncologists can effectively cure pretty much, to generalize, half of all cancers. That wasn't the case 30 years ago. But the only people we're interested in is the half where we treatments don't work. And that's the big challenge in oncology, is knowing what to do when all the standards don't work. Right now, there are 3 or 4 revolutionary therapies out there, but they cost anything from $50,000 to $1 million to treat an individual. So they're not standard things yet. But there are incredible treatments coming through that have amazing effectiveness on cancer, various cancer types, when all else has failed. And out of ethics, you don't treat a person with an experimental compound or idea, even if it's a safe one or an expensive one, until you've gone through the standard list. And a lot of people don't have that time. You can't wait 3 months to see if chemo has been effective for some people. So this is kind of the pain and the pressure point for us to do something about that. I don't know if that answers your question.
I think it was awesome. It was awesome.
It does. Yeah, totally.
I'm going to ask one more question about AI. And then I think we can have a break.
Still right?
That's okay with you, Phil? What role is AI playing, do you think? Or do you know?
Artificial intelligence has a huge role to play in proteomics in several ways. To talk about AI and ProCan, I've explained that there is around 8,000 proteins we identify in every tumour sample, and we've got 28,000 of them. So you can picture how many proteins we have in our database. A human eye can't analyse that data in the way that we need to. So AI is used extensively now in trying to compare proteomes across thousands of samples. And without it, we just can't do that on that scale. We've recently completed our first study based on AI, very specifically able to look at a subset of those samples and predict the tumour type and predict where the tumour may have originated, the tissue of origin. It's an extraordinary effort, and it's just the tip of the iceberg for how important machine learning and as a part of AI is going to impact on the ability of us to use our data to reach our goals of predicting treatments for individual patients. It's truly revolutionised what we can do. Another example, AI has impacted the basic study of proteins itself in incredible ways. So many of you are aware that the Nobel Prize for Medicine went to a group who are machine learning scientists who invented a process called AlphaFold. And this is pure, simple AI. And it takes the 3-dimensional structure of a single protein and compares the shape of one protein to all other proteins. Going backwards, there's a database of the structure or shape of proteins. Proteins start out as strings, but the strings fold into 3-dimensional shapes. It can be that. And knowing those shapes is critically important for understanding protein function and drug design. So AlphaFold came along and said, well, there's a database now of all known protein structures, and there are hundreds and hundreds of them. Can we predict how a protein will fold from a string into 3-dimensional space just from the string, just from the amino acids that make up the protein's fundamental structure? And it works, and it's unbelievable science. That was AlphaFold 1. Then they improved it to AlphaFold 2, and as we speak, AlphaFold 3 has come out that can predict now where drug binding sites are on the structure of a protein. To put that in context, it takes years to identify and determine the structure and shape of one protein. And then they get put into a public database. What AlphaFold is able to do is take the gene and the list of the sequence of a single protein and predict a shape for proteins where that data, experimental data, doesn't even exist. And that's incredibly powerful as a research tool. It's certainly deserving of the Nobel Prize that was awarded, because it's synergizing researchers like me who are trying to understand drug binding sites or how proteins interact with each other. There are some proteins for which we've never learned the structure yet, and we can predict them, so we can go on with the research about what this protein is and how it works. Thinking of each protein as a single molecular machine. That's the best way to look at a protein. They're machines that do things. And they're machines that build more complex parts to assemble. And they're machines that work as single individual units. Proteins are amazing.
On that note.